Hello everyone, my name is Farial Sharif, a student in Dubai National School, grade 10B. Today I will be discussing the topic of proteins and amino acids. What is a protein, you might ask? Well, it is basically the workers of the cell. They do almost everything. Naturally occurring, extremely complex substances that consist of amino acid residues joined by peptide bonds. Example of protein cells are collagen that creates your cellular structure, or actin and myosin that produce muscle contractions, or also enzymes that help in speeding up chemical reactions. Proteins are made of amino acids, or in other words, the monomer of proteins are amino acids. Each amino acid has its own chemical and physical properties. There's 20 different amino acids that have 20 different R groups. Some are polar and some are non-polar. Some are charged, some are small, some are medium, some are bulky. But these properties in turn determine the properties of the protein cell. Proteins synthesize and break down. Dehydration synthesis binds two amino acids together, forming a dipeptide, shown below. A long chain of amino acids is called a polypeptide. Hydrolysis separates dipeptides and polypeptides into individual amino acids. Polypeptides fold up into proteins. A chain of amino acids folds into a unique 3D shape to become a protein. The function of a protein depends on its shape or tertiary structure. Proteins, primary and secondary structure. The primary structure of amino acid is a sequence of a polypeptide. Secondary structure, which is also known as a substructure, is localized areas of coils, sheets, and loops within a polypeptide. The tertiary structure of a protein refers to the overall three-dimensional arrangement of its own polypeptide chain in space. It is generally stabilized by outside polar hydrophilic hydrogen and ionic bond interactions, and internal hydrophobic and interactions between nonpolar amino acid side chains. Quaternary structure is the overall protein shape, arising from interactions between the multiple polypeptides that make up the functional protein. Only proteins with multiple polypeptides have a quaternary structure. Denaturation. Proteins are therefore vulnerable to conditions that alter their shapes. Heat, excessive salt, or the wrong pH can denature a protein, changing its shape so that it can no longer carry out its function. As an example, consider what happens to an egg as it cooks. Heat disrupts the hydrogen bonds that maintain protein shape. The proteins unfold, then clump and refold randomly as the once clear egg protein turns solid white. Typically, a denatured protein will not renature. That is, there is no way to uncook an egg. Here are some questions to test your knowledge. Pause the video and answer each question based on what you've learned. Once finished, correct your response. Now, you will have to compare your answers with the answers shown here. The first question is, what is a monomer of a protein? At the beginning, as I mentioned, proteins are made out of amino acids, which make amino acids the monomers. How many types of amino acids are there? There are 20 different types with 20 different R groups. What is changing? A protein shape called. It's called denaturation. Why are proteins important? Proteins are large, complex molecules that play many critical roles in our body. They do most of the work in the cells and are required for the structure, function, and regulation of the body's tissue and organs. Thank you for watching, and I hope you have gained a benefit from this video.